all that they've been submitted back to the X Prize. So we're really in a holding pattern now for that. Fran has also been working on the bioinformatics side of things, and we'll have something to share properly with everyone soon on that front. It's more like kind of taking all of the work that we've done on the bioinformatics side and putting it into libraries, Python libraries that other people can use.
Mm-hmm.
It's pretty cool.
Yeah. So later during the call, we're going to talk about like working groups, about how we could actually bring the community together, about common interests, subjects in the community. And Martin, you raised actually a point about how do we move forward with some of the project that needs like distribution so do you want to maybe just raise the like in 1 minute your little point, and then we can invite the community later for a more serious call together.
Sure. Well, I think that with a lot of these open concepts and open source projects that are going on around the world, in the biomedical world, it's very difficult to translate something that's actually going to impact patients because of the liability side of things, getting access to clinical data, how much it costs. But a big part of doing any of this is when you talk to the regulatory authorities, you have to have an entity or a body that's applying to the regulators. You can't just say, oh, we're a load of guys on the internet, They're like, oh, we don't really know how that works. Not yet. If we figure that out, we'll have done a great thing. But we ultimately need to figure out how we're going to leverage all the work that we've done to allow regulatory bodies like the EMA and the FDA and each national body to kind of work with us in a way that they understand, to move the work that everyone has done and the incredible work that people have done all around the world and turn that into a product that clinicians can engage with and that regulatory bodies can engage with, and then that ultimately can be manufactured and delivered to patients. And that's something that no one has really done yet. And I think trying to figure that challenge out would be really worth investing quite a lot of energy into.
Mm-hmm. And so the idea would be that we start actually like a brainstorming group about how can we create something at the JOGL community level that can really help all the projects that are going to go through that step. What form can it take? Do we have to create a special entity that can really help do that and basically be there for more like the more person to apply for those community projects, etc. So we'll be sharing a date for a future meeting where we'll be discussing specifically of that. So keep updated, stay in touch. Thank you, Martin. I guess we can start with the normal schedule. I'm going to share my screen. So first of all, welcome to the global community call of the OpenCovid19 initiative. I believe this is the 25th one. So quite a long way. Here we go with the screen. Is it working? Yes. Here we go. All right. So this is better like this. All right. So just for newcomers here, those are like the main links if you want to interact with the program later on. But first, general announcements, news. We just talked about the fact that we're opening some new community working groups. We'll also be having another discussion with Tatiana later on about a similar one. But first, also, I'm super excited to introduce some new JOGL team members. We're joined by 3 amazing people who are going to introduce themselves. Kate Rushton, Paige Perila Piratuan, and Gamili Adzaro. I guess, Kate, you're first. Would you like to introduce yourself?
Hi, I'm Kate. I'm a UX designer and I'm going to be working on JOGL Spaces and the general JOGL platform. I've got a work history in open innovation as a community manager and strategist.
Awesome. Thank you, Kate. Paige?
Hi, everyone. I just joined JOGL at the beginning of this week as the outreach ambassador. So we're going to be working on those questions that Thomas talked about at the very beginning of the conversation about how do we make things easier for the community to engage by themselves on the platform. And I have a background in animating local communities in East London, actually.
Thanks so much, Paige. And then we have Gamili.
Hello, everybody. My name is Gamili Ajao. I come from Ghana. I live in Accra. I'm currently working with JOGL as a program manager for JOGL Africa. I'm 1 week older than Paige in JOGL. And so far, I've worked across public health, education, and also building open communities. So yeah, really excited to also connect with Open COVID-19 community and learn more about what's happening within this space.
Awesome. Thank you so much, Gamili. So let me share my screen again. The first subject that we want to share with you, the fact that we are actively preparing for a crowdfunding campaign dedicated to the Open COVID-19 initiative. The objective is plural here. We first there is a big need to raise awareness about what this community has been doing, and so far there is little, to compare to how good a crowdfunding campaign can raise, awareness and also create a great dynamics. So the idea is to not only raise dynamics, but also raise funds for the projects, because there are still a lot of projects that are in need of funds. And so far we've been only raising money in a very centralized manner through JOGL and our partners. And so that's been working out so far, but it's really, not very much a community-based philosophy. So the idea is how can we introduce also a way for the general community to also not only support projects by their time and skills, but people who maybe would not be able to contribute directly to projects because either don't have the time or because they necessarily don't feel like they could actually contribute by their skills, that they could also do it because they associate themselves with the goals and the mission of the community by providing some funding. And so that was like the missing piece so far within that program. And so this is going to initiate a new kind of dynamic for JOGL, for Open COVID-19, and for this community. And obviously, the idea is this is going to benefit directly to you, like to your projects, because like a big chunk of the money that's going to be raised is going to go directly into macro grants. And so we'll be also opening a working group about the organization of this crowdfunding campaign, because we really want to co-develop this campaign with you all, so that it serves really the purpose of this community, and that we can really work with you all in terms of sharing and animating this campaign, so that we can all benefit. And so we have some ideas about what kind of perks could be relevant for this kind of campaign. We thought about it could be actually you sharing some of your time to present your projects, or making, providing a visit of your lab, or stuff like that. And the idea is really that to as much to provide you with visibility for what you're doing, celebrate you, and as much connecting, being able to, for the people that are outside these kind of practices, practice communities, to understand better what's going on by meeting you and hearing from you. And being able to understand who they are going to support with their funding. This is going to be also very helpful for helping JOGL to raise more funds, also because this is going to, again, raise the dynamics and the awareness about this program, and so to attract more organizations that would like to donate more. So the first meeting for this working group is going to be on Friday, this Friday at 6 p.m. CET. And so if you're interested to contribute, because either you have already done a crowdfunding campaign or just because you're curious and you just find the idea, again, good and exciting, you're very, very, very welcome. This is going to be quite an adventure. The idea would
Martin Kodar.
Sorry?
Sorry.
Sorry,
I'm
it's okay. The idea is to launch it in a month or two. The idea is to be quite fast because the momentum is right now, and the project also needs awareness and funds as soon as possible. So yeah. For those who are interested, see you on Friday. You'll be hearing from us on the different channels, for the community mobilization. Next is a call that has already been done previously by Camille. We are currently harvesting feedbacks from the OpenCovid19 community so that we better understand what this program has been helping you on, what has been good, or what has been actually frustrating also for you, so that we can improve what we do. And so this is very precious for JOGL and our future capacity and capability, to support you guys. So we'll be super appreciative if you could spend, it's just a few minutes, the time to answer this survey. So thanks very much in advance. Then we have been given also the information that there are a certain number of people who can't make this global community call because they are not in the right time frame. And so we've been thinking about how we could have a double date, like a double time. One would be the one we have currently. Another one would be another, like something that would make the one living in India or in Australia being able to join. So it's an open question, and if you're concerned by this time scale, please just contact us through the Slack and tell us what would be actually the best time for you. One proposition would be to do it around 10 a.m. CET, so that would be 2:30 p.m. New Delhi, 5 p.m. Manila. It seems like a good compromise, and so that we still have the people in Europe, in Africa that can join, with the one in Asia, in Oceania, and the one at 6 p.m. is still good enough so that we have the whole American continent we can participate to. So it's going to evolve. We have to take some decisions here, and now we're going to hear about some of the projects that are bringing updates. And some announcements. We are going to also have a little discussion about one of the working group that Tatiana wants to raise awareness about. So first, I don't know if for the project that wants to provide an update, I hope that you actually put your name here, or maybe didn't. Let's see. Okay, we have Hackett 19. So Sophie, would you like to start?
Yes, why not?
Oh.
Hello, everybody. Just a quick update for us. I had a talk with my team a couple of days, week ago, to see what they want to do and where we stand. And our developer told me that for him, the app is okay. It's responsive now. The team is pretty busy. We are all busy anyway. So it's difficult for them to continue to push the project. However, they would be glad to make a presentation of the tool. So we just have to find the right date to do it. And well, what we would need would be to have help on making the promotion of the tool and find maybe some sponsors or maybe a company who would like to push it further and make it usable at a larger scale. So this is where we stand now. It's in French, in English. I think that I still have 1 or 2 modifications to do, translation modification to do. But yes, it can be used now. And it's really glad to see it working properly on the mobile phones. We used part of the grant to pay the trainee who made the site responsive for us. So it's good for us and it's good for her as well, because it was a good experience for her to work with the team, an international team, and to experiment making a site responsive. So she was very happy about that, and if we can make the call with the team, she will be with us.
All right, thank you so much, Sophie. The next project is Corona Detective. Do we have someone from the Corona Detective team? So let me share my.
Actually, I'm also from Corona Detective.
Okay, awesome, Tatiana.
Yeah, but I'm not really in all the chemistry stuff the guys are doing, because I'm involved in bioinformatics and all this informatics and analysis related stuff around. So I know they are working. I know that guy has his samples and he's working on this, but unfortunately I cannot tell you details.
So we have here the slides. They're still working on the X Prize. They've made a small funding request. Actually, they got the funding through the micro grants.
As I know, Rachel already out of I believe buffers or something like this, like some chemicals already since weeks. So we already spent some extra money from budget of Aquarium on this.
Okay.
Theoretically, would be nice somehow to close the gap.
Okay.
But again, Rachel, she knows better about all this situation.
Okay, so we wait for Rachel to arrive. I believe she may actually join later. The next project is a basic respirator with Hunter, but I haven't seen her in the call. So she last time she updated us on the fact that she updated also their app to make a scan of your face so that you could adapt very well her mask, the mask that she created on your face. So I encourage you to go and check her project page on JOGL. You have more information there. Then we have the Bioreactor project with Adrian. Didn't see Adrian also in the call, so I'm just going to read what he's written. They continue work on integration. They enhance the software by fixing bugs and add new capabilities. They're changing the casing by moving to a stronger material and redesign. Great, and exploring new sensors. That's awesome too. And of course, always great to have more resources. Yeah, I hope that all those projects that are in need of more resources will be able to join the effort for making the crowdfunding campaign successful. Then we have the Rapid Action Patients Dispatch, XCOV19, that is being led by Akur. And so I don't know if we have someone from this project in the call. I believe not. So here again, they have a website also that you can check, My COVID Connect. So I'm probably not going to read everything, but basically, if you're interested in a software like a platform for enabling logistics to facilitate mass scale patient testing, you should definitely check this project. So it's project 251 on the JOGL platform.
One thing they need in particular, if you are connected to any people in India, especially people working in hospitals, they are looking for interviewing, like doing 30 minutes interview with people working at facility, because what the technology they're developing is trying to be like one of the target audience are facilities and patients, so that the patient can be directed to the closest facility to be tested. So if you know anybody, feel free to share. We're also contacting our network to support them. We never have enough help. So yeah, feel free to share that in your network.
Thank you, Camille. Then we have Accessible Genomics. So it's a project to facilitate genome sequencing and diagnostics, also using DNA amplification. And so it's a project 285 on JOGL. They have a verbal agreement from the Philippine Genome Center in Danao to collaborate on the project. It sounds great. So they need more team training with the Philippine Genome Center, I guess also need to order more equipment in regions and crowdfunding. So I guess again that's going to be very synergistic with what we're going to put together. So we'll be talking with Khalil. Then we have the 1 hour COVID test using LAMP. I don't know, so if Sarah could join, probably not, because she said she's teaching. That's a project 163 in JOGL. They have already completed the blinding proficient testing of the 157 samples that were sent by the X Prize, and their result looks very promising, so that's exciting. They're still waiting for the feedback from the X Prize, and they're also waiting for the green light from the APHP to send their kits for clinical validation. Right now, the APHP is a big hospital cluster in Paris. They've been a very good partner to JOGL and the OpenCovid19 initiative so far. They're also, one of their laboratory is testing the kits, the diagnostic kits being developed by the community. Right now, because of the COVID situation in France and in Paris in particular, they have actually stopped any research, scientific work, and have all focused their equipment and resources on just performing tests with existing tests. So this collaboration is going to be rebooted once the situation is going back to normal, at least a more acceptable level. So probably not before at least 2 or 3 weeks. All right, and then we have new projects, I believe, that would like to introduce themselves. We have the CoronaNet research project with Cindy and Vanessa. So I will leave the microphone to you.
Okay, awesome. Thanks so much for organizing this. This is such a great initiative, and I've really enjoyed learning what everybody else has been working on. So just to introduce myself briefly, I'm Cindy Chang. I'm a political scientist by training. So I'm a postdoc at the HFP Technical University of Munich. And with me is Vanessa Spiesel. I actually don't know if I'm pronouncing it right.
Spiesele, it's Swabian, so Don't bother.
Still learning German. So she's a project study intern working with us on this project. And I thought that, just in this quick introduction, I'd just say a few words about who we are, how we're doing what we're doing, and what our needs are in the coming months. So the Coordinate Research Project has been collecting data on government policies made in response to COVID-19 we've been collecting the data starting in late March, but the data, the data itself goes from December to the present day. We have almost 50,000 policies collected, which makes us the largest group doing this. And it's also, we collect the data in a very fine grained level of aggregation. So we have a bunch of different policy types, so including lockdown policies, external border restrictions, mask wearing, health testing. There's in total 18 broad types and more than 100 subtypes. And we also collect data on who the targets of the policies are. So are they asylum seekers? Are they prisoners? Is it, and also the geographic target, is this a policy that the national government in France is making towards a particular province in France, or is it something that applies to all of France? So this is the type of information that we've been collecting. The data has been available publicly since early April, and it's our methodology and everything is on GitHub. Just to say a little bit more about how we're doing this. So the first step is just figuring out what policies there are to code. And so on that end, we have partnerships with a couple of companies that have been using machine learning algorithms to scrape newspapers and government policy documents to sort of give us the raw data to work with. But the real sort of challenge is that all of our data is actually hand coded. So we have more than 500 research assistants who then look at these different documents. They also go to their own sources, and they document these policies in a survey that we designed in Qualtrics to standardize and centralize the collection of the data. So the survey asks the research assistants all the same questions for each policy, and then the results or the responses are then standardized. We communicate with our research assistants using Slack as well, so it was cool to learn that you guys also use Slack. And this is how we sort of get feedback from them, and we have lots of internal structures at this point, like regional managers, country managers, data cleaners, data validators, who are kind of working on different parts of the project. When a policy is then entered, there's a data cleaning team that comes and makes sure that it's properly coded. We also have a data validation team that, so we sample 10% of the data, and then have it triple coded, so that we have 3 independent coders for the same policy, and we can then assess if 2 of the coders reach the same conclusion, then that policy is valid. If they disagree, then a 4th person comes in to resolve this discrepancy. I can post, so we have our paper on the data set was published in Nature Human Behavior, which provides more detail as to the methodology there. So this is sort of where we are. What we sort of need in the coming months is more support in terms of data validators, data cleaners, regional and country managers, people who can help us with visualizing the data. So we've been really focused on data quality. So one thing I will say is I think that our data format is pretty difficult to use. Right now it's an event data set. And ideally we'd like it to be more flexible so that it can be a panel data set. The issue is there's so many dimensions of the data. The question then becomes, well, what panel do you then want? So we kind of want to make it as flexible as possible. We have been really lucky in that we've gotten an EU Horizon 2020 grant to help us continue this project, which is really great. And it's sort of like a mixed blessing, because it keeps me and it keeps like one of our PIs employed for the next 3 years working on this project. But we still will need to rely on volunteers for like most of the actual like data collection and data clean. Like it's just, I don't think there's anything near enough money in social science to really properly compensate people for their work. And yeah, it's kind of unfortunate because, like, for example, we had a great team coding subnational policies for Nigeria, and we had Nigerians coding their own policies, like what was happening in their country, and a lot of them had to stop because it was getting too expensive for them to pay the Wi-Fi and the electricity to continue working on the project, which is just like kind of heartbreaking. So now we have to recruit new people who likely won't be from Nigeria to code policies like on Nigeria, and it's just sort of these are the sort of challenges that we're facing. But yeah, this is the end of my spiel. Happy to answer more questions if you have them.
Well, thank you so much, Cindy and Vanessa. This is a really a brilliant project. I believe one thing that I could see how that could help you. This morning I was in discussion with one of the coordinators of the innovation network of the WHO, and so we're in discussion about how we could help each other. And so I'm going to present some of the project of OpenCovid19 in December, at the 17th of December, to the whole innovation network of the WHO. And so I believe that that's typically like the kind of project that could be highlighted, because it's touching exactly what they're working on. They have local coordinators of the WHO, and so they could definitely help in local regions about, you know, in trying to get access to data and connecting you with local actors. Otherwise, I've been also contacted today by there are more and more boot camps, you know, for data science, teaching people about how to do data science in general, and they're always looking for opportunities to test their skills. And so I was just asked this morning about if we had an interesting project that we could submit to them. And so I believe that's typically something we could connect you to.
That would be excellent. Thank you.
Yeah, otherwise, great. I agree that social sciences are usually very much underfunded. It's what you just said is really heartbreaking. And you said, like, not being able to pay your own Wi-Fi. Well, what's going on? Do you have? I believe it's. Oh no, we have actually another project. So before we go to Proteus Presso from David, do you want to react to what has been just said by Cindy and Vanessa? I guess the discussion will move further in the Slack. So thank you again. Happy to have you join the community, and I hope that we'll be able to support you and help you achieving what you are aiming for. Then we have David with the Proteus Presso project. David, the floor is yours.
Yeah, thank you. Hi, everyone. The Proteus Presso is intended for a second wave, what we call it, for helping other projects like a Bioreactor Project. There's one project that is intended for synthesizing the most precious molecular reagent that we require now for COVID-19 protein detection that are essentially enzymes. The problem is that this process is actually very expensive normally. So we are trying to figure out other alternatives in order to produce those enzymes at a much lower price, at a much lower, and it could be accessible to almost anyone. So I had a vision ProteoPresso just when I realized that my handheld handheld presso machine was broken. So I said, well, maybe I can take a few pieces of that and try to experiment how we can make a mini version of an HPLC. HPLC is one of the bulky machines that are required for the protein purification. Those machines can be extremely expensive, and a few labs actually have them. And in order to produce in an industrial level pipelines protein for the detection of COVID, so it's a very strenuous, a very complicated process. So we decided to simplify that. So I started in parallel where we started working with the bioreactor, testing some prototype using even Lego machines, Lego monitors. So it crystallized into another machine that would help us for purifying the protein. So the idea is using another molecular biology approach. So we have plasmid that normally produce our proteins that currently they have like the his-tag version is a very common version of tagging protein for purification. But the column that's required for his purification can be extremely expensive. So we decided to remove the tag and substitute for another that could be much cheaper. So we're using another one that is called CAR9. It's a similar method for, instead of using this extremely expensive Sephiroth metal-based chromatography, we decided to move it into something that could be much cheaper. Instead of using that, we decided to use silica. Silica, like a smash glass, you know? So the machine is going to be similar to a handheld hand presser machine, but probably a little larger. So we are working now in selecting the best material that we can incorporate into the final design, and we're also working on the PCB board. It's going to be controlling on the function. So essentially, we'll have like 2 pumps for processing the sample. So after the sample is being sliced, it's going to be purified into several batches. So it's going to incorporate a fraction collector, and it's going to incorporate some pumps and automatic pumps controlled with an app. So at this moment, we have, well, actually I have it with me. We have the first version of the fraction collector, but this is, well, the toy version. It's actually pretty bulky, but it's going to be downsized into something much more smaller, like probably this size. And we are working at this moment on the molecular biology for substituting the original construct with the new construct, so we can instead standalone purification of the protein with that method. And hopefully, at the end of the project timeframe, we will be able to deliver the first prototype. It could be functional prototype that we incorporate a dedicated PCB, the dedicated electronics, kind of the first application for controlling with the mobile devices, and a molecular construct that it will be like the first construct that we're going to test using proprietary silica column that we're going to build, of course. Yeah, so those are the things that we're working at this point.
That's great. Do you have any specific needs that you'd like to share?
Actually, I have some slides of a few of the things that we have already accomplished. So maybe it's a little bit quick. That this is from another construct that we prepare for the yeah, let me see. Oh, I think it's not. Okay, can you guys see my screen? Okay, so this is one of the molecular strategies that we have. This is the previous result that we have. Sorry?
Could you go full screen, like presentation mode?
Oh, okay.
Yeah, just play, I guess.
Okay, yeah, it's a little bit slow now.
Okay. If you can make it into 2 minutes, that would be great. We have another subject of discussion afterwards.
Yeah, okay, here we go. So this is on the left side, you will see like this is the molecular biology strategy. So in the left side, you're going to see like the plasmid that we already have prepared. And the second is the one that we intended to achieve. So we're including into one of the already tested DNA polymerases that we have achieved to purify it using a traditional method. But we're going to substitute the tag for using, instead of the his tag, with the CAR9. And we are still discussing, we're not sure at this moment it will be convenient or not, to include a TEB protease site. So that TEB site is for helping for removing the tag. The problem with this is kind of perhaps it might be an issue, which we're not sure. The problem with adding CAR9 is that you're adding a larger chunk of another protein. So that could interfere with other activities, like for the, I don't know, maybe the processivity of the DNA polymerase. But of course, that's the reason we are testing the two purification methods and the activities. Let me see, this is more or less like the idea. For the binding material that we are proposing two very cheap and affordable material. One is silica, the same silica that you use for reducing the amount of humidity in a room. And the other one is titanium oxide. Titanium oxide is extremely cheap as well because it's used for the creams on the sunscreen formulations. So those are affordable, and there are already demonstrated evidences that those can be used for actually capturing those proteins using this CAR9 and peptide. So this is more or less like the protocols and standardization that we want to include into a database that's going to be included as part of the Proto application. So we know that each protein is going to be different. We're working particularly on these enzymes, but we believe that other enzymes are going to require several changes. So for each of the numbers, there are going to be like several revolving methods that are going to be modified accordingly on the final application that we're going to incorporate into the machine. So, well, we have already this valuable equipment and the things that we need to order. And we can more or less separate like in 3 areas of development. One is the molecular biology for the substitution and the standalone purification steps. The following is the hardware. Then we're still in the process of measuring which could be the most affordable. And we have help from other people. We are receiving help also from Eric Mars and Fadel Samati, that they're the founders of the Proteopedia. Fadel is actually one of the leading crystallographers in the world. I had the chance of working with him when I was a postdoc in Japan. And well, together with Eric, they founded Proteopedia, but also including some of the protocols for crystallizing the protein. And also that was very useful for choosing the right protocol that we might include for the purification of several other proteins. And also as a kind of a more technical help, it's a friend I have that worked for Thermo Fisher that will help us with a few of the formulations that are already available. So yeah, so this is a little bit of what we're doing at this point. The prototype is still like on development, although we have like a proposal, like it's gonna be a kind of a canister isolated with some foams, and the electronic is gonna be placed on the top of it, and we'll have like wireless communication capabilities. So yeah, we are happy that we have some designers that are proposing very sleek and nice ideas, although we think the reality is going to be a little bit different. Maybe it's going to look like more like the early version kind of look more like an espresso machine. But well, we're happy with the idea being proposed. So, yeah.
Thank you so much. Yeah, it looks promising. Bioproduction is definitely one of the key steps, in order to make biotechnology having an impact. So it's still very much a bottleneck. Crossing fingers for you, and let us know how we can help along the way. We have a few minutes left before the end of this call, and I believe that Tatiana would like to raise also a point for how to help this community move forward when it comes to legitimacy and maturity of projects, getting recognition. Tatiana, would you like to?
Yes. I could, of course, introduce the idea. So it's raised in our Corona Detective project, and it was an issue because I have some acquaintances working at Nations in the decision prevents department. And once I just wanted to tell them, like, we have here tests, COVID tests, they are cheap, they are good, they are nice. Would you like to try them somewhere? And response I got, are you certified? We cannot do anything what is not certified. And no one would take a risk to use something not certified in such a huge bureaucratic organization as Nations. With this situation, we were discussing kind of several meetings in Corona Detective, how we could organize something. We raised some acquaintances of Katrin Haddad in Germany, who tried to help us to clarify the whole certification process. But nevertheless, we never came to any kind of clear workflow. Then the same idea appeared, as I know, also in the USA, and with kind of, coming through the same problems, we could see that there is also no clear workflow, because they're like, we need a lot of people which are not usually somewhere around like a biology-inspired open source community. So it's rather more work for some kind of lawyers, for some kind of maybe politics, standardization, whatever, working people. Yes, and what would be good to have, it's a kind of like probably institution, probably some kind of standards. I cannot really now to shape it properly, but any kind of organization or procedure which could provide certification for open source projects. And what I'm thinking, why this is important, it's like in our time when all this, let us call it technical and social achievements becoming so horribly distributed, we also need infrastructure for outcome of all these horribly distributed communities. And now what we have, it's only centralized, and there's like 2-3 huge centralized points like FDA in USA, like the European Certification Commission, which from many sides, like complexity of procedure, like which exactly like how the project should look like and who should be responsible for it, just doesn't fit to current state of art here. So now we have kind of like a new social, let us say, appearance of open science, but we don't have any kind of institution to prove feasibility of this open science. I mean, it's just like, if you ask some common human what we are doing here, we are just hobbyists. And we just, in eye of probably like a common human, we have nothing to do. And because of this, we spent our free time with this. And like, you know, again, from outside, we are not reliable. But here in these projects, I mean, of course, there are some projects which are not very serious and not very interesting or not very with outcome. But nevertheless, there are also projects which could be kind of like, you know, which could accelerate evolution, kind of evolution. And like there's a potential. And for now, there's no mechanism to make this potential like, you know, spreading in society. And yeah, so shaping all of this, we were like already talking around in different groups with different people in different circumstances that we need any kind of trustworthy approval of things which are done by open source communities. And for this, yeah, we also were chatting today with Thomas that it should be something. Probably we have to set up a group. Probably we could set up an institution, satellite institution of JOGL, because some people, as I've noticed, which would like to have any influence on this, but they don't want to be part of JOGL because of some political reasons. We could actually, how to say, leave this, skip this enthusiastic people. So, and because of this, probably it should be like, as I already said, satellite of JOGL, not a really working group, because we were observing a bit such situation yesterday. I mean, I'm absolutely open to any kind of thoughts, any kind of ideas considering this, because it's, again, it's a new situation, which I don't know about humanity, but me personally, I've never seen before. And it would be very nice somehow to, you know, to wrap this idea, at least with some kind of milestones. Somehow, yeah. Okay. I mean, of course, we could start as a project group, as an initiative, as something. But as I said, because this is completely new, and because every person who's interested in this movement worth for now for us, because it's not a lot of them, we should somehow organize it this way, which could leave satisfied everyone who want to contribute to this.
So thank you so much, Tatiana. I think it's also a very core subject. It's one of the core missions that also JOGL is trying to achieve is to provide legitimacy for such community-based initiatives, and so that we can act also as an intermediary body you know, with institutions, so that like what we're doing with APHP, for example, and some others, and or foundations that wants to fund research. And so there is definitely a need to move to go even further. Like, how can we even structure this in a way that we could even imagine some kind of standard procedures for doing like a more thorough evaluation, or even like provide like some kind of status that could be recognized elsewhere. So that, you know, I think being able to work on this collectively is definitely needed. If you agree, we could make a proposition of a first meeting, probably like next week, to establish some kind of what are the main objectives, what can we achieve in a short time, what can we improve at least, you know, in a very short time with what we have, and establish like some kind of foundations, you know, to make the subject evolve, because it's key if we want to generalize this way of, you know, community-based science and innovation. This is necessary. Like, we have to be able to provide some kind of guarantees, you know, the same way as, you know, in the corporate world or in the institutional world, they need also some guarantees, so that you can, you know, it's about trust basically. And so we need to be able to provide, you know, a trustful relationship to others, and it's normal for them to doubt, you know, what these kind of communities and projects would be doing. It's normal, it's natural. So we need to counteract this, with way that can be understood by everyone.
Yeah, I completely agree with your point. So yeah, I will be happy if you announce.
Yeah. Let's do that. Let's discuss together about the day that could convene both of us, and we can invite whoever actually can participate. We'll make an announcement in the Slack of OpenCovid19, and the idea would like to have like the first meeting probably early next week. That would be great.
Okay.
So I just jumped on. I'm sorry, my class just ended. But yeah, I mean, I'm kind of just at the end of this, but I mean, I think it was really interesting, Tatiana, and maybe I'm reading wrong what you were saying, but I think it would be interesting to have a physical location, if possible, for JOGL, or call it JOGL, or call it something else. But I think that actually I never thought about it before, and I don't know if that's what you were even talking about, but to have like an actual institution with walls that is like, in addition to our online platform that we're participating in right now, for some reason, I think it would go a long way to have a research institution, just because it's called a research institution. And it's a physical building, and that then we could be collaborators or, work at this institution. It's all about like, it's not about fooling people, but it's all about like, these regulators, they're used to a certain format, and it's somehow we have to bridge that. And so, of course, it's only in the US, but I'm very active in talking to lots of groups of people about the idea of an institutional review board for the biohacking community and for open science. Like I'll be talking next month to some clinicians and some clinical researchers and some people on institutional review boards about how we should hopefully convince them that it's important for them to consider broadening their scope of who is a valid scientist and what is a valid research institution. And yeah, anyway, so I'm very interested in this idea, and sorry to jump on late.
No, it brings a lot to the debate. Thank you so much, Sarah. No, you're absolutely right. One thing we could do, I think your proposition is very relevant, is so far JOGL has walls also. Like, we are based in Paris, we're in the Center for Research in Interdisciplinarity, which is a research institution. And but we could say that JOGL also has walls within old laboratories who would like to somehow host JOGL locally. So you could, like, for example, your laboratory, Sarah, could host like some JOGL activity, in a sense that you could actually be the local correspondent, for the JOGL community, and interact with local actors, with the legitimacy that we can provide with the host, that the whole system provides, basically. So having a distributed approach probably would be even more efficient, because if we centralize everything in one location, I'm afraid that we are going to lose the kind of agility that we're looking for here. So if we can somehow still have maybe as you said, because we need to have like a simple way to talk to those institutions. So maybe have a HQ address, but that is just symbolic, and then we can register associated addresses in different parts of the world that corresponds to existing laboratories, like community lab, for example, and that can apply basically also in the name of the general community. I don't know if that makes sense, but that could be a way also. Yeah.
I'm sorry, I have to jump to another one call.
So let's
Actually,
make the group picture before. So we have a tradition that we make a screen capture and we just say hi to whoever is going to watch this video afterwards on the social media. So I'm going to make a screen capture. Are you ready? All right.
Yep.
Say hi. All right. So we're going to announce the date for the working groups. I think that Sarah would be great if you could join. And in fact, what you just talked about, the fact that you're so proactive, talking to those clinicians and those institutions to create some kind of review process for the biohacking community, that is so important. And I'm sure we can work together on something here also. So yeah, I'm looking forward to those great elements. I guess if anyone wants to add anything else, maybe.
Yeah, okay, sorry, I have to leave.
Yeah, bye-bye, Tatiana. I guess that's it. This is the end of this call. It's been super nice to hearing from all of you. Thank you so much again. And well, look for the announcement that we'll be sharing, especially looking like regarding the working groups and the upcoming crowdfunding campaign. Thank you so much, everyone.
Hi to lots of new people that I don't.
Yeah. We just welcomed 3 new members in the JOGL team also. Yamili, Kate, and Tej. So you'll be hearing from them.
Welcome.
All right. Have a great
Bye-bye,
evening,
everyone.
day, morning, wherever you are. Bye bye.
Bye, everyone. Nice to meet you all.